Well, today it's about the factors which influence foliar absorption of nutrients. It is, I think, a common practice to use this, especially in orchards, if you think about micronutrients or calcium sprays. I do research on this stuff for many, many years. I started rather theoretical, and now I moved to, as you can see, University of Applied Science. So I'll focus more and more on application of that. And I want to give you a brief introduction into what we know about this process of foliar absorption. Well, the structure of my presentation, first I want to give you an overview of the general characteristic features of foliar. It's very different from root uptake, and I want to emphasize that. Then I want to show you where the uptake takes place. This may be rather theoretic, and maybe you may think it's not interesting, but it is, because if you want to apply and we know where the absorption takes place, maybe we can optimize this process. And finally, I want to talk about the main factors controlling the efficiency of this whole process. So how can we optimize uptake rates? How can we get all the stuff inside? And what are the factors controlling that? It is, first of all, the type of salt we are using. You can use different type of salts for applying, for example, calcium, and the environment. And the most important factor there is the humidity of the air. So let's start. What are the features of foliar fertilization, and what are the main differences from soil uptake? Well, everybody using foliar application knows that it delivers the nutrients quite fast. It's a fast absorption, fast supply, and the main advantage is then when root uptake is limited. Maybe if root activity is low because the soil is too dry, or the pH is so high that the plants cannot absorb, for example, micronutrients such as iron. And one advantage is that we can save time and labor and money by applying it together with, for example, pesticides. It could be mixed in the tank with the pesticides, but we must be aware that the uptake takes place by an organ of the plant which is not designed for it. The organ which is thought to absorb the nutrients and the water are the roots, and this poses some difficulties. The main function of the leaf surface is, on the opposite, to protect the plant. There's a barrier. We have the cuticle on the right-hand side, which is a sort of water repellent skin, and this reduces water losses. It prevents the penetration of, for example, pathogens or stuff which is not intended to enter the leaf. And this also applies for our nutrients. So we apply the nutrients to a surface which is mostly intended to protect the plant from penetration, uptake of other material, and this poses the problems. So if you now compare plant nutrient uptake by the soil, we have the uptake under more or less physiological control. Roots can actively absorb nutrients, and they can reject nutrients, they control it. So there's a selectivity, which is not the case when we apply it to the leaf. There is no physiological control. It is a simple passive physical process. It is diffusion, and the leaves have no way to select which material enters in which concentrations, which is totally different to the roots. And this is the main problem, the main challenge in this business is we have the hydrophobic surface, more or less. It depends on the species, but generally they are more or less hydrophobic. They repel the water and the nutrients inside. And our nutrients are hydrophilic. They are water soluble, they are salts, or they are water soluble like urea or boric acid. They are dissolved in water. And the second is, of course, what I mentioned already, It is a passive process. It's diffusion. It is only driven by the concentration gradient, which means if you have a high concentration on the leaf surface, it will at different speeds, but it will definitely enter the leaf. And now the art of foliar fertilization is to achieve the optimum uptake rate. Optimum means enough to supply the nutrients to the leaves. So, example from my research, iron deficient corn in that case, it's not an orchard, it's corn. I applied iron chelates foliarly, and what I ended up with iron toxicity. So there are many ways to do it wrong, and therefore it is important to know how it works. Well, where is it taken up? What we apply to the leaves? There are basically two main pathways. The first is the cuticle, the skin, this water-repellent skin, and the stomata, these openings in the leaves which allow the plants to absorb carbon dioxide for photosynthesis. So this is a simple model of the cuticle. We have the outermost cellular layer, called epidermis, which is covered by the cuticle. The lower part of the cuticle, the so-called cuticular layer, contains cell wall material. It is attached. The cuticle is glued to this cellular layer, and the cell wall parts, they help to fix it there. And the upper part is thought to be free from cell wall material. It contains the pure cuticle, so-called cuticle proper. Well, what is noticed when we study the permeability of the cuticle now for salts, that it is strongly dependent on the air humidity, relative humidity. Victoria Fernandez from Spain and I, we reassessed the published literature, and we wanted to find out how the permeability of the cuticle depends on humidity. And what was found by analyzing a range of studies is that below, let's say, 80% relative humidity, the permeability is very low, and it only increases if air is moist, if humidity approaches saturation. If we have 100% humidity, for example, only then we can have high humidities. So how does it come? People think that the cuticle may contain water. The cuticle may swell and shrink. It may absorb the water. And here in this scenario, we have dry air. We have the droplet with applied nutrient, for example. It rests on the cuticle, but it cannot move because there is no water in the upper part of the cuticle. The water comes from the epidermis. The epidermis contains water-containing cells, and the cell wall contains moisture. So we have water only in the upper parts of the cuticle and not directly underneath the drop. This changes if the air becomes humid. So we have a high humidity. The water can also be absorbed from the atmosphere, and the water then is also present in the upper part of the cuticle, and this may form such pores where the nutrients may diffuse in and may enter the leaves. And this model explains why the permeability of the cuticle is so much dependent on humidity. So this was the first uptake pathway, which is well known. The second one, the stomata, has been under debate for many, many years. People doubted that stomata play a role. I studied this in my PhD thesis, and we provided some evidence that it actually works, that stomata may contribute to foliar uptake. So one of the latest milestones in this process of showing that stomata play a role was with nanoparticles. So we did not use nutrients, not ordinary dissolved tracers. We used particles, and the idea is those particles are so large that they cannot enter the cuticle because they are solids. They are about 40 nanometers in diameter, and they can only enter the leaves via the stomata, and we are able to show that this actually takes place. So this is a cross section where the surface has no stomata. You see the epidermal cells and the material is on the surface. And if we made this optical slicing of the leaf, and we, by incident, we sliced in the middle of such a pore. These are the guard cells forming the pore. You can see at the light microscope. And this is the stomatal pore. And we found that the material entered into the leaves using these stomatal pores. They migrate, they diffuse on the surface of this pore. So in the light microscope, we can see, or translated into the microscope, the material moves here through the stomatal pores. But this does not take place with every stomatal pore. This is a different experimental system where we studied where this dye, this is a fluorescent dye, where this enters the leaf, and we found a way to visualize where the material enters. So these dotted lines indicates where the drop was located containing this dye, and these red dots show where it enters. So it enters not everywhere, only some very specific points. We still don't know where and why, but what we can say is stomata play a role, and they may play a very important role, but not every single stomatal pore can be penetrated. And this can also be found in real life. So this is an example with peach leaves, which were previously chlorotic because iron was lacking, and the iron-containing solution was applied to the leaves. And in early stages, we can see that the regreening takes place, but very spotty, very patchy, only at single spots. And this quite nicely fits together with our results with this fluorescent dye. So to summarize this, this is a schematic view taken from the textbook of mineral nutrition, Marsh's textbook. We were asked to rewrite this chapter, Victoria Fernandez and I, and we developed this sketch. So we see the epidermis containing stomata. Here are the guard cells, the specialized cells forming the stomata. And here we indicate with the arrows where the foliar material, the foliar applied material can be absorbed. It can go through the cuticle, but there is a quite high resistance. It can also go somewhere here probably. So in the close to the guard cells, close to the stomata, but there is a very potential pathway when stomata take place in this process, then the material can enter along the pores. And here you feel like at the stomatal pore, the right-hand side, this should indicate that not every single stomatal pore can take place in this process. Okay, this is in a nutshell what we know about foliar fertilization or foliar uptake pathways. Now let's go to the main factors which control this process, which cause high or low uptake rates. It's the ruling factor is, according to our results, the air humidity, but also it depends on the choice of fertilizer salt. Usually we have a range of salts. We can we can apply calcium as a chloride, as a nitrate, or as a formiate, or whatever. There are different types of salts available, and the question is, how may this affect uptake? So if you now have a look at the processes on the leaf surface, we must be aware that the changes, that the concentration of the foliar applied nutrient in the solution changes. Usually we apply quite dilute solutions. The salt concentration is not that high, but as soon as it is on the leaf surface, or even before, even in air, this may, while still moving in the air, this may happen. The water will evaporate, and the result is that the concentration will increase. This is displayed here with the increasing intensity of the yellow. It's getting more and more concentrated, and it may reach a point of maximum saturation of the salt. Every salt has a limited solubility in water, and once this solubility is exceeded, the salt will start to precipitate, and it may eventually dry out completely and be present on the leaf surface as a solid. it's important to be aware that uptake can only take place as long as it is liquid. So here, the substance can be absorbed. If it is solid, if it is a dry residue on the leaf surface, it cannot be absorbed anymore. Well, it can be revetted, of course, for example, by dew. When humidity increases about a certain point, it may become wet again and uptake may continue. And the question is now, when does this happen? So when does the solution dry out completely? When does it stay wet with a high concentration? So in terms of uptake rates to be expected, the highest uptake rates are to be expected in this situation. We have the maximum concentration on the leaf surface, and according to the laws of diffusion, this will result in maximum penetration rates. So the question is now, does it dry out completely? It depends on humidity. If humidity is high, it will probably not dry out. If air is dry, it may probably dry out. But this also depends on the type of salt. Every salt has a certain threshold, a certain limit at where it dries out, and this is called the deliquescence point, or relative humidity of the deliquescence, or point of deliquescence. So this is the humidity where the salt dries out completely, and every salt has its own distinct deliquescence point. It depends a little bit on temperature, sometimes more, sometimes less. Potassium nitrate, for example, strongly depends on temperature. Other salts are less temperature dependent. A little bit, so we have to take it into account. How are these deliquescence points? I show you here some examples for calcium salts, for potassium salts, for magnesium salts, for zinc salts. And you can see that the range of deliquescent points may be quite large in the case of the calcium salts. So almost saturation with this is formiate. This is a calcium salt which is sometimes used for foliar application. This is the chloride, 31%. The range may be lower with other salts, for example, with the potassium. The range is between almost saturated to 85, but we can see a general rule of thumb. The chlorides always have the lowest deliquescence point, sulfates the highest, and the nitrates somewhere in between. Okay, so what does this mean? This means that we have to expect lowest uptake rates when the deliquescence point is high. So this is what we tested this in a short experiment where we used faba leaves, which is quite convenient in the lab. So we made an experiment at 45% humidity, and we're using the sulfate, which is dry at 45 humidity, because the deliquescence point is somewhere around 90%. The nitrate has a deliquescence point just above 50%, and the chloride just around 30%. So the expectation is high uptake rates with the chloride because it remains dissolved the whole time during the experiment. We may expect a bit lower uptake rates because in the final state, when it is in equilibrium with the humidity, the solution will have been dried out. And we expect very low penetration rates with the sulfate because it has a so high deliquescence point that it will dry out quite quickly. And expectation result fits. So this is the absorption rate. We have more than 75% absorption of the applied magnesium. When we applied it as a chloride, we have a little bit above 50% when we apply it at nitrate, and we have below 20% if we apply it as a sulfate. Conclusion may be, oh, don't use the sulfate. It's not good. The penetration rates are so low, but we have to be aware magnesium belongs to the macronutrients. We need high amounts, and therefore, if high amounts would penetrate quickly, this would probably result in leaf damage. And this is actually what happens even in this trial. So as a chloride or as a nitrate, penetration rates were obviously too high. So the result was we had this leaf damage. Okay. Every salt has its distinct deliquescence point. This can be seen here in this graph. We see here the concentration of these nitrogen salts in this case as a function of relative humidity in equilibrium with this humidity. And you can see here, just as an example, that deliquescence point of ammonium nitrate obviously is just about 60%. This is 0.60%. But why I show this graph is, it is quite amazing, and maybe people are not aware, when humidity increases, the concentration in the drop, in the solution, decreases. It approaches zero. In theory, in saturated air, the concentration in a drop would be zero because the drop absorbs so much water that the concentration keeps on dropping. So intuitively, we would say, oh, the higher humidity, the better, but this is not true. And we were able to show this in a quick experiment with coffee leaves. In this case, we applied calcium nitrate to the upper or the lower leaf side. And what we found in both cases, that we had maximum penetration rates at intermediate humidity. If humidity was too high, uptake was quite low because the concentration on the leaf surface actually was too low. And if humidity was, on the other hand, too low, we also had low uptake rates because the solutions dried out on the leaf surface. Okay, one final remark. If you see these tabulated values of deliquescent points, etc., be aware that this only applies to pure salts. As soon as you have mixtures of salts, some contamination, that the deliquescent behavior of the solution changes, and this is particularly pronounced in this case here. Urea ammonium nitrate solution is used as a nitrogen fertilizer. The single components have deliquescence points of almost 60 or above 70. The mixture itself has 18% relative humidity. Okay, I have limited time, and I'm afraid I have using too much time already, but I'm already at the summary. So both the cuticle and the stomata are important uptake pathways. So this refers to where is it absorbed. The second is the deliquescence point, together with the relative humidity of the atmosphere, are the main factors controlling this process. Substances with a high DQ, in our example, it was magnesium sulfate, may be taken up slower, but this may render it more compatible because uptake rates are not too high and leaves are not damages. If humidity drops below this DQ value, uptake ceases, and at high uptake rates, at high relative humidity, we have to be aware that uptake rates are quite low because the concentration in the solution is quite low. And my final remark was that you cannot take the tabulated values of deliquescence points and transfer this into your situation. You have to be aware that your solution may be not pure, it may be a mixture, and then you cannot use the tabulated values of deliquescence. Well, that's all for now. I thank you very much for your attention.
Thank you, Tom.
I stopped the presentation, and here I am.
Yeah, thank you, Tom. It was a very interesting information, and I think all our specialists will find it very interesting and use it in their approach to fertilizer. And it once again demonstrates that fertilization is becoming a high intelligent technology. Thank you again, and hope we will see you soon in Moldova, or you'll come here.
It would be a pleasure, but we have to wait until it is possible.
Sure,
if it is possible, it would be a pleasure to meet you, visit you.
Yeah.
Thank you very much.
Thank you. Have
Bye.
a good time.